Hi, I'm Ian Donaldson. In this video I'm going to talk a little bit about the data consolidation process that is used by iRefIndex. In the last video I showed you that iRefIndex build database consolidates data from about 10 different interaction databases. I also showed you that we started with about 1 million protein-protein interaction records, but only ended up with 500,000 or less than 500,000 distinct interactions, meaning interactions that involve the same two proteins, regardless of how they were shown to interact or what kind of interactions they had. In this talk, I'm going to explain why it is that there's so much redundancy in the data and how we actually dealt with that. So the problem of data integration is shown here. If this represents a simple interaction record between two proteins, and I've shown the n-ary gene official symbols here, this interaction may be defined in the intact database. However, it may also be defined in the bind database under a different interaction record. The problem comes when looking at these records on the surface. Both of the databases may use different accession systems to describe the proteins that are the interactors in these records. Bind will use the refseq system or some GI system from NCBI, while intact will use the uniprot KB system. So what we did was we went through and pre-processed all of the accessions that we could find in all of the interaction databases and retrieved the sequences for them. And for each sequence, we've calculated an SHA-1 digest, message digest, and this results in a 27-character key. We do this for all of the interactors that we find in the database, and then are able to determine which interactors and which records are actually talking about the same thing. If we do this for the entire database, we're able to resolve which interactors in which records are talking about the exact same sequence from the same taxon. What I haven't told you here is that this key also takes into account the taxon ID. We also, of course, keep the original accessions, the refseq and uniprot KB accessions. So the solution is based on an exact sequence matches between interaction records. The actual keys that we use, the ROG IDs and RIG IDs, are an extension of what's shown here. A ROG ID is this 27-character key that's the digest of the sequence, and that gets appended to the taxon identifier to create the ROG ID. In a similar manner, we make distinct keys for every single distinct interaction called RIG IDs, or redundant interaction group identifiers, and that is also a 27-character key that's formed by ordering the ROG ID keys and then redigesting with the SHA-1 algorithm. And then we end up with a view like this in iRefScape, and you can see under the Edge attribute browser over here, the iRig ID is the same for these two entries, showing an interaction between the two proteins I just showed you in both bind and intact. So what I want to do now is go back into some of the implementation details to talk about how we record provenance and the way that we make these mappings. So if the large black square represents a PSI-MI XML record given to us by one of the interaction databases, and the two smaller squares inside of this represent interactors A and interactors B, we look at specific pieces of information for both of those interactors, including the primary ref, the secondary references, taxon identifier, and the sequence if it's available. And here's some examples of what those look like. So I'm just going to concentrate on one interactor because we do the same thing for all interactors that we find in all interaction records. What we've done is we've pre-calculated a table that contains a lookup of all of the database accession pairs and the expected taxon identifier with the SHA-1 digest key appended to the taxon identifier, and that's the ROG ID. Then what we do is we simply look first at the primary reference to see whether or not we have this in our table. If we don't find it, we go on to the secondary references. So in this particular example, it's quite an easy mapping. We know that this corresponds to this ROG ID here, and it may also correspond to interactors in other records as well. So all of these things inside of this circle are referred to as a ROG, or a redundant object group, things that are interactors that all have exactly the same sequence from the same taxon. And in this case, it turns out to be n-ary gene 7132, or one of its splice isoforms. Now, In the process of doing this mapping, we also have an assignment score, and that keeps track of the operations that we went through in order to get to this assignment. In this case, it's very simple. It's a P, meaning primary. We looked at the primary reference. We found it in our database, no problem. This assignment score is associated with a binary bit flag where there's about 17 different things that can happen through our workflow, and each one of these bit flags gets flipped when one of those things happens. And in this case, it's a very, very simple process, and the human readable version of this is simply a P, meaning that the primary accession was used. However, things can be much more interesting. In this case here, we have a human readable score. The assignment score is PTU plus O, meaning that we started with the primary accession that was provided to us by the interaction database. However, we noticed that the taxonomy identifier that was given by the interaction database didn't match what we expected to find. We tolerated that, but we changed the taxon identifier that was associated with the interactor. The primary accession that we received was outdated, as indicated by the U here, and this is probably a uniprot accession that needed to be updated. And when we tried to update it, it turned out that there were multiple possibilities that we could update it to. This is a secondary accession that becomes demerged into two different sequence records. However, it turned out that only one of the possible sequences that we could update to matched the ROG ID of the original protein sequence that was given by the interaction database. Okay, so this is a screenshot of a figure in the original BMC Bioinformatics 2008 paper where you can read more about the details of the assignment process. But basically, the take-home message is that these assignment scores are associated with a path through this workflow. The results of the entire process of doing the consolidation are also described in the paper. I just want to show you this one particular matrix, which is a comparison of the overlaps between all of the databases. And in this case, in this particular version of Bind, there were 62,903 interaction records in Bind in total, but there was an overlap between BioGrid and Bind of 23,000. These numbers down here represent the number of interactions, distinct interactions, that were found only in those databases. So the take-home message here is that all databases contribute some distinct interaction records, and for that reason there's a need for doing this kind of data consolidation to find those records which are distinct. Everything that I've talked about today can be found on the iRefIndex.uio.no site. The link to the paper is given in this section of the web page, and I will try in just a second to show you where on the iRef website you can find these mapping scores that I talked about. If this is an iRef interaction that we're looking at in iRef web, and you click on the proteins that are involved, these are the original source records that show an interaction between these two things here. If I expand this section here and I scroll past all of the aliases that can be used to describe that protein, right down to the very bottom, I come to protein interaction mappings and assignments. And you can see in the OFEED record, they provided us with a primary identifier for this interactor that was actually used. We didn't have to update it or anything. The final mapping that we mapped it to was exactly the same thing, and it gets a code of P. But for HPRD, they gave us some other identifier that we didn't know about, and so had to rely on the secondary identifiers. The secondary identifier in this case was exactly the same thing that OFEED gave us. However, they said that there was a taxon associated with this of mammalia. That's not what we were expecting. We were expecting Homo sapiens. And so we updated that to Homo sapiens and gave the T code for the mapping assignment score, telling you that the taxon had to be modified. Okay. So just back to the talk. In subsequent talks, I'm going to talk a little bit about representation of complexes by iRefIndex, and then give a brief introduction to iRefScape, the Cytoscape plugin from iRefIndex as well as the iRefWeb resource. Thanks for listening.